Good morning, everybody. Today, Mike Martin, who's our MPC this month, is also our speaker. It's a big day for Mike. He introduced a new member, and he's a speaker, and he's an MPC. And he was recently elected a trustee as well. Now, his career was in the pharmaceuticals. And you know that New Jersey is a center of pharmaceutical research and business. This industry is one of the most regulated and also one of the most innovative and consequential to all of us. Just consider the drama of the COVID epidemic. The acid test of a new drug or therapy is clinical research. And Mike was right there for a 40-year career, running clinical projects of all sorts, including one that has to do with RSV, for which a vaccine came out this year, which many of us may be getting. Mike is also an entrepreneur. He has a patent for a COPD therapy that he may tell us about. His talk today will be about pioneering discoveries, drug development, licensing, and what happens when things go wrong, and much more. Please welcome Mike Martin.
Thank you, Mark, for that generous introduction. Okay, how this talk evolved, I got to this point. I gave a presentation at the Reminiscence Committee at the request of Bill Tittle. And essentially, I talked about the development of a therapy, not a vaccine, a therapy for respiratory syncytial virus, which I licensed in. We partnered with Gilead. Quite frankly, had it been successful, I'd be establishing the Monaco chapter of the old guard. It was not. So we'll talk about those failures later on. In addition, I also participate in the investment forum, which DJ essentially hosts for the old guard. And there was a number of queries on biotechnology, when I should invest, and this type of thing, which I won't go into in any detail in this presentation. But as a result of that interest, basically, I thought it might be appropriate to talk about this topic. It's rather broad, essentially, but I want to make it relevant. I specifically selected compounds, which many of you may be on now, and how they were discovered, how they will work. Not all of them, I obviously don't know your medical history, but again, the idea is to make it relevant. The second point I wanted to make was essentially that I talk about human interest stories about the people behind the discoveries, how serendipitous this particular activity is, despite all the processes, science, and everything else that goes into this. So it makes for interesting human interest stories. So that's essentially where I'm going to take you, talk about process, discovery, key products that have been developed, and I'll throw in, if there's time, some information on the Inflation Reduction Act. Okay. First of all, in terms of my background, I essentially have a large pharmaceutical experience. That was my background. I worked for several companies, essentially through consolidation. People went on, much the same if you're in financial services. That's what essentially happened in the industry. So most of it was with Sanofi, Roche. I worked for Merck for a while. So it was in really two functions: licensing which is essentially the acquisition of technology from third parties. That acquisition involves clinical development. So in other words, you go out to company X, you like the compound, it fits with your therapeutic strategy, you bring it in-house, you pay the money for that privilege, and essentially develop the compound. These particular transactions can either range from $50,000 to upwards to billions. So obviously, in those cases, I'm not the key decision maker, the CEO is, but essentially it relies on science, business, and a number of other factors. From licensing, I went into clinical development, which I mentioned was symbiotic with essentially licensing. There's clinical development activity as part of that. So that's essentially my background. And I have background in many different therapeutic areas because licensing took me into different therapeutic areas. So, and this presentation will cover several therapeutic areas. My principal area of essentially business activity and development was in virology and oncology, and that's it. So what is the outline of the presentation is where do new drugs come from? How do drugs work? What are some examples of how these particular drugs work? So I'm taking it from theoretical to practical, so you understand the full continuum of essentially logic here. Interesting to get it to the human side, what are the types of drugs that have been developed? I would call them eureka factors, essentially drugs, that glimmer of hope in a lab. This is talking about penicillin, a number of those compounds that were discovered by prominent scientists that have won Nobel Prizes. The other factor, which is not well known, are entrepreneurs that essentially, with a very limited experience, licensed in products that essentially what I do and develop them. And essentially, I know some people in this room that basically are on these compounds. And I'll talk about some failures in my own licensing activity to give you the full kaleidoscope of essentially my work. And then finally, I'll talk about essentially the Inflation Reduction Act. Okay, so it's the process of research, the sketch of interesting discoveries, discuss pharmaceuticals that are relevant to you, and how drugs are essentially repurposed for new uses. Now, again, the two functions that I were involved in is where do basically new product ideas come from. Again, as I mentioned, I was involved essentially for 30 years in external activity, licensing products in from a variety of institutions. At the other extreme is essentially internal development or discovery within the pharmaceutical company themselves. Generally, companies focus on a tight focus of therapeutic areas, and essentially that varies from company to company. For example, in licensing, the sources of new ideas come from small companies, essentially the NIH and universities. Most of these types of, with regard to universities and the NIH, they never quite make the threshold of proof of concept that a pharmaceutical company likes. I used to receive in telephone books from major universities on chronicling 120, 150 discoveries that they had made without much clinical proof that essentially these things would work. The proper audience for that particular subject matter was really venture capital, which is further downstream in the development of products. Some of them did, but it was a very difficult, essentially, evaluation. And for the most part, essentially, these were pretty much discarded, with the exception of a few that have applicability to a particular therapeutic area that we're working on. The business issues generally regarding licensing is that they're more expensive than internal development. You have to pay the company an upfront fee, and a variety of milestones to essentially secure the rights to the product. Essentially agree with the company on what the clinical plan is. Believe me, this is not something that's easily decided. The small company is interested in essentially proving the drug works. For example, in oncology, I'll just throw out an example. Somebody may be interested in bladder cancer. A large company is going to be interested in lung cancer because it's a far bigger market. So these decisions have to be grappled with. Who pays for what is the direction of the clinical program? And to be honest, looking at essentially these issues, it takes about 6 to 9 months to resolve these issues. Intellectual property is involved, who pays for what is involved, what type of deal structure is involved. These are all complicated discoveries. Discovery is a much easier process. It's an internal program. You have certain expertise, whether it's oncology, virology, cardiovascular care, but you're focused on essentially those particular therapeutic areas. The priority among these programs are essentially set by management based upon risk assessment and the beneficial impact, and the total control and less expensive. While I've painted these two subject areas as symbiotic, it's quite confrontational. The issue is that a budget for development is set forth by essentially the company. And licensing, which brings in a compound, essentially has to tap into that budget of research, which means internal research, has to drop some projects that some gifted science has worked on. I used to characterize my function as the man who stuck his head through the canvas that people would throw organic matter at him. So that's putting it politely. Because there is no way licensing essentially could outgun internal research in terms of scientific expertise. We had our own scientists that went along with this particular proposal, but that was essentially, so what happened, many of these decisions, not all, went strictly to the CEO. He resolved essentially the priorities where they would be. That was determined by global needs, whether research was productive for that period of time. Did this give us a leg in on other areas? So there's a number of issues involved, patent issues and the like, but I won't get into the weeds on that stuff. But essentially, it's a very challenging process. Just for example, I did 30 transactions in a 30-year career. We looked at perhaps 100 licensing ideas a year. So it took maybe 10% seriously, most discarded. So that was essentially the way it worked. What types of pharmaceutical products are developed? Now, this is getting down to the nitty-gritty. It's meant for people that basically have no understanding. There's really two types: small molecules, oral. Many of you are on blood pressure medication or arthritis pills, this type of thing. These are synthetic molecules that essentially are developed. At the other end of the extreme are large proteins. These involve essentially monoclonal antibodies, other proteins and biologicals like vaccines. So the two different buckets. And why am I pointing this out? They're of course administered through different means. Generally speaking, oral compounds are oral. Essentially biologicals are infusion or injectables or subcutaneous injections. Now, this is important because people talk about cost, and the direction of pharmaceutical research is in the large molecule area. That's where the discoveries in research are heading, and away from oral compounds. And there's a number of reasons for this. Science is driving these decisions, but also essentially there is longer exclusivity regarding essentially a protein, a large molecule, than an oral substance. Generic competitors essentially can knock off pretty much most oral compounds. They have great difficulty with essentially knocking off a large molecule or protein. Why? The investment in the plant, as I'm talking about, essentially, you can add a tablet press into any pharmaceutical plant and produce another compound with proper quality control. In the case of vaccines, large proteins, essentially, you need a dedicated plant. The cycle for the size of the investment would vary from 20 to 30 million for essentially a new oral compound for a generic company, 500 million plus for a large molecule. Most generic companies, A, will take the risk that they'll get approval for that particular compound, and B, essentially won't allocate those resources to essentially. So the direction of the industry is basically in the direction of large molecules, not always, but most. Now, what are these large molecules? To make a concept clear in your mind, if you see these monoclonal antibodies, they're usually MAB at the end of the compound, or essentially a protein, Humira, Skyrivi, all these compounds you're being inundated with advertising. These are essentially large molecules that fall within this bucket, are very difficult to replicate. Enbrel, for anybody on arthritis, rheumatoid arthritis, that's where that's used. The product I in-licensed, that is on the market to this day, which is about a $12 billion product that essentially, how would you say, we thought was about a $500 million product when we licensed it in. So it tells you we could be wrong. We were wrong on basically what the indication for that product would be. The manufacturing process for these compounds are quite different. You can see the cost involved. Essentially, you can essentially produce an oral product in 3 months. For a biological, take you 6 to 9 months. So the manufacturing time and investment, consequently, the cost of these compounds is considerably more, and the supply is much more at risk. The cost of the therapy for oral, and certainly many of you are on compounds that probably cost you $5 for a 30-day supply. By contrast, some of these large molecules, you're talking about $100,000 a year. And the reasons are, there's barriers to entry for that particular compound, the nature of the research, and quite frankly, as the last indication, there's very little generic competition for large compounds because of the investment required to establish manufacturing. And I won't go into the difference in clinical trials. That's another story. Okay, now let's talk about basically how drugs work. This was pioneered, essentially theory, the Nobel Prize winner was a German chemist by the name of Emil Fischer. And this governs essentially the way research is done today. Essentially, he saw cells as essentially a number of receptors on a membrane of a cell, which was essentially, in this case, I use the metaphor of a lock. That's the receptor. The drug essentially is the key which finds that lock. Now, there's a lot of issues involved with how much the drug basically is affinity to that particular receptor. But this is basically the way molecular biology is conducted today. And that's essentially, you have an understanding of the biology of the disease, and you have a product which you will design to essentially fit that particular receptor. So that's conceptually the way you think about pharmaceuticals. There's 2 types of basically drugs that basically fall into 2 buckets. They're agonist, which essentially initiates an activity in a cell, or an antagonist, which prevents an activity in a cell. So they bind to us, they're the lock, they have different attributes, the drug, whether it enhances an action or inhibits an action. More graphically, I put this in a traffic light to make sure the concept is crystal clear. An antagonist stops the action. An agonist lets the action continue. The side effects are, of course, the wrong receptor, and we'll get into that. The ideal product for a pharmaceutical company, at least according to Fisher, was essentially the RX hits the exact cell receptor. That's essentially what, so in other words, you've identified an activity that emanates from that particular cell, and you design a product that hits that activity. We all know that's not the case. Many of these drugs may hit that receptor, but they hit other receptors which cause side effects. Now, the most, I guess, prevalent example of this is in oncology. To give you an idea how oncology has evolved to this day, I would characterize it's gone from what I would metaphorically call napalm therapy to stealth bombing. And by that, I mean, essentially, most of these historic drugs were used to essentially inhibit cell growth. That was the target. The scientists and clinicians had very little to work on. To give you an example of therapy that was used historically in this area, and is still used, mustard gas from World War I. That inhibited cell growth, was used up until recently. Arsenic used. Essentially, cisplatin, still used today. It's a metal compound associated with platinum. It's still used today. So these are, generally speaking, chemotherapeutic agents. What we've migrated to is essentially these large molecules that essentially are more targeted. And that's essentially. So what happens when you take a chemotherapeutic agent? It essentially kills rapidly growing cells. What happens? Your hair has rapidly growing cells. Your gut has rapidly growing cells. So hence you talk about hair loss and essentially nausea and vomiting associated with these particular drugs, and also suppression of your immune system, which, because of its inexact nature, which is targeting the cancer cell itself, this is kind of peripheral or collateral damage, and that's essentially what's happening here. So they are hardly exact in terms of their targeting, but they were effective in their day. They're still being used to a large extent. So what are drug interactions? So from the Fisher guideline that I presented, essentially we're talking about two drugs hitting the same receptor. And you can see how one particular drug would interact with another, inhibit its ability to essentially cleanly target that particular cell receptor. Some examples. So going from the theoretical to the practical. I know a lot of you are familiar with the weight loss drugs. It's a panacea for everybody in this room. However, these are essentially agonists, and what they do is basically they enhance insulin flow, which essentially metabolizes sugars. So essentially you're losing calories through that activity. They're agonists. In this case, I cite an example of opioids. Essentially, they bind to a certain receptor. They mitigate certain hormones involved in sensory perception, and basically soothe pain. And examples of that. Here I talk about, which I know several people in this audience are probably on, are high blood pressure medications, metoprolol, a varieties compounds which had LOL in the suffix. These particular compounds work, essentially their objective is to reduce high blood pressure and also work selectively in essentially treating congestive heart failure. Now, what do? They attach themselves to essentially the beta receptor in your heart, and they essentially prevent adrenaline from hitting that particular receptor, thereby slowing down your heart, and are also receptors on your vascular walls to essentially dilate your blood vessels. Other compounds that work by this, which are, there's no women in the audience, but in the case of breast cancer, most breast cancer works, the therapy that works, at least up to date, is essentially drugs that block the estrogen receptor on the tumor. Now, most 90% of breast cancer is basically estrogen receptive positive. And what the drugs do is block that estrogen. So it blocks the vehicle which will grow the tumor. So again, another example of an antagonist. Now, I'm going to turn to the process of developing drugs, and this is pretty obvious. It's told to me very early on, in God we trust, all others have data. So there's nothing more than essentially an academic scientist who thinks essentially he has some key, he may have a key to it, but essentially it's a skeptical audience because you have to prove these drugs work. It is more than an insight. One fact, one clinical trial can upset an entire theory about essentially any number of therapeutic theories about treatment of particular people. Let me go on here. I went back. What is the criteria for FDA drug approval? 2 well-controlled clinical trials, essentially where you show efficacy and tolerable side effects. These can be conducted in the United States or overseas. They're essentially agnostic about the opinion where the trials started. But essentially, these criteria have been waived in certain circumstances. For example, in the 80s, where AIDS was a particular problem for principally the gay community, the FDA approved drugs that had 1 clinical trial. I can't remember the data on it, but essentially the idea was the FDA was convinced because the mechanism of action to essentially control the AIDS epidemic was well-founded in virology theory. It's the same theory that governs essentially the use of Paxlovid as the treatment for COVID. It's the same theory that covers essentially the treatment of other viral disorders. And what essentially is that? It blocks cell receptors. A virus goes into a cell, okay, tries to go into a cell and replicate. What essentially these particular drugs do in AIDS and basically COVID, Paxlovid principally, is they block the receptor. They're an antagonist and prevent the virus from essentially taking over the cell. So that's basically what's going on here. The trials were double-blind, placebo-controlled. All right. What is the criteria for clinical proof of efficacy? Well, there's primary endpoints, which is essentially what the FDA and the companies agree to, and there's secondary endpoints, which establish. Generally speaking, you have to hit the primary endpoint to get the drug approved. There could be several primary endpoints, but for simplicity, I'm just going to talk about a few here. And I'm going to go through examples. In the treatment of stroke, for example, these substances in your blood system that are called platelets, which essentially aggregate as you get older. Conceptually, to think about it, it's rust in your plumbing. That rust has to disaggregate to prevent stroke. And essentially what happens here is that that is a secondary endpoint. You have to show that that drug works in that capacity. It will not, and I repeat, will not be approved unless you show it actually prevents stroke, either ischemic stroke or hemorrhagic stroke. Likewise, in prostate cancer, which I'm sure we're all aware, we get our annual PSA test. I actually had a drug we were developing in this particular area where we showed the definitive proof that the drug reduced PSA levels in prostate cancer patients. It did not show statistical significance in reducing the size of the tumor. FDA said, go back, do the studies again if you want approval. So it sounds intuitively obvious to us, but the FDA will not accept improvement in secondary endpoints or biomarkers in lieu of quantifiable evidence that the drugs work. Now, in osteoporosis, for example, the original drugs were approved with essentially reduction in symptoms. Osteoporosis is really a female disease. And essentially, the FDA changed its standards and basically went to the prevention of bone breakage, which is a natural evolution of this disease. Now you say, well, what do I care? Now, let's just think about this for a second. We'll go into the cost of these studies and essentially how one drug's approved and a cost of basically, if you're trying to show within a population of osteoporosis, and somebody here may know the answer to this, but let's say 20% of the people basically will have bone breakage. So you can enroll 10,000 people, hoping to basically identify 20% that have breakage. It is that 20% which you'll compare to placebo. So let's talk about numbers here. Generally speaking, the cost of a clinical trial, is about $20,000 per patient per year. Getting to the point, if we're talking about the cost of clinical trials and the intended effect on drug prices, to prevent bone breakage in a population of, say, 10,000 people with osteoporosis, perhaps 20% of them essentially get a broken bone. So for $20,000 a patient, for 10,000 patients, it's $200 million a year. Just to essentially identify a subcategory of 20% at $20 million a year. And it's for 1 year. So you see the incredible cost involved, and not only a risk. You don't know if you've identified the population where indeed the drugs. So when you talk about cost of development, it's absolutely gone insane. Okay. How are modern therapies developed? Now I'm going to go through all of this, but what is the organization for a clinical trial? You essentially have an RX company, a large company with money. That's essentially how it's done. You identify a drug candidate. You essentially set the objective, how many patients you're going to essentially, you consult with the FDA, what the objective of the trial is. You have an independent scientific board, which essentially can halt the study. They're outside essentially the purview of the company. So why is the study halted? It's either halted for superb efficacy. Novo Nordisk just announced that it halted its trial for its diabetes drug, which showed improvement in kidney disease among diabetes patients. So extraordinary results, quite frankly, they halted it. On the other hand, you have certain side effects which evolve, which I don't really have to go into, but they're halted for that reason. Now, who does the actual work essentially in conducting these trials? Basically, it's a contract research organization. They identify the clinical sites where the drug is actually going to be tested, the number of patients they can recruit, and in some cases, for example, just to give you some war stories, if you're dealing with, for example, blood pressure, no problem. You can get thousands of patients into the trial, blah, 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 blah. But let's say you're dealing with Gaucher's disease, or Pompe disease. There's 2,000 people perhaps in the world that have this disease. How much you think it's cost to recruit a 30-person trial with Gaucher's disease or Pompe's disease or any one of these obscure diseases? It's probably close to $100,000 per patient. And the reason for that is that A, you don't know, you have to find these people. You have to get them to a facility that knows this disease. You have to find experts which are few and far between, and most of the time outside of the FDA, who understand that disease. What are you after? Symptoms, progression of disease, any number of factors. But the difficulty in doing these trials, I can't say, it's really, to the average person in here, probably doesn't really appreciate that, but that's what's done. We had one trial for an orphan drug disease. We flew patients from Europe to the United States every month. You can imagine the cost of doing that. And essentially the drug worked, so fine and dandy. Who is involved from a functional discipline? The disciplines on the left are all involved in one stage or another in drug research, or in licensing for that matter. But essentially the ones I've outlined in red are the principal people that basically talk about, are essentially involved in the actual monitoring of the clinical trials, the planning for it, and essentially the oversight. The rest is really at various stages. Okay. Now we'll go through this. I'm going to go through this quickly to give you an idea. Drug development process is essentially a process, as they've indicated. I try to outline essentially the probability of success as you go along here. It's kind of like a funnel, so to speak. But if you're in preclinical, the chances of something, this is essentially characterized by, you have an idea that there's biological activity, you have a receptor identified, you have a drug candidate. You have perhaps information on animal studies, you have various other non-species type information. There's probably a 5% chance that that drug is going to make it through approval. Then you move along into phase one, probability starts going up. The principal objective here is to basically talk about dosing. So your idea is you want to understand the maximum tolerable dose among patients where side effects erupt. Now, how do you translate from animal studies to human studies? Interesting question. You take an animal species, you weigh it, and essentially you translate that effective dose into a human being. You know, a dog weighs 20 pounds, you'll take it into a human being, and essentially with 5 times the dose at a certain weight level. Moreover, or else you'll take the dose depending upon the size of the patient, his height and stature, and translate that from an animal-based study into a human study. So essentially, as we go along here, you'll see patients, we go from test tubes to essentially greater and greater, essentially, patient involvement as the drug passes thresholds through essentially efficacy and side effects. The business items there are basically listed. I'll just blow through the objectives. Basically, it's safe and effective, the maximum dose in the phase 1 studies, dose selection, what is your final dose, or at least what you think your final dose selection is, and did you hit the clinical endpoint, as I described before. Now, a little bit more granular, the probability of success for something in preclinical, this varies. There's a dispersion around this 5%. I would say, to take an obvious example, Alzheimer's disease is probably close to 1, close to 0. Antibiotics is close to 20%, because in each case, animal studies are predictive of antibiotic success or dermatology success case, whereas in CNS and complicated disorders, animal studies are not particularly predictive of activity in human beings. So your understanding of biological activity, essentially the patients we've described before, cost, et cetera, et cetera. I'll blow through this. Here we are in phase 1, where there's a 10% probability. Essentially, we've defined the expected efficacy based upon that study. We have maximum dose, essentially exposure to the patients. There's 10 to 30 patients. We forecast business, and essentially we establish the dosing. Now, I just would divert for a second to the people who are interested in investing in biotechnology stocks. I personally don't invest in anything prior to phase 3. And I'm just going to give an example of this. At Roche, we licensed in 30 compounds from preclinical to phase 2. Now realize we're talking about a group of rheumatologists, oncologists, clinicians, neurologists, people with expertise. Not one ever made it to commercial development. Now, I'm just going to handicap anybody in this room with that statistic. Be my guest. But for me, I want to save my money. So just a word on that order. And the other item, just on investing, if you're interested, just to divert for a second, you have to invest in a company which is cliched with good management. Now, what does that mean? That means that that CEO has actually developed a drug in that therapeutic area. That qualifies, in my judgment, as one objective criteria as to why you should select that company. There's too many academicians who are around who great scientific credentials, but great science doesn't translate into clinical benefit. And I think that disconnect has to be really understood by the people in this audience. Okay. So then we're in phase 3, final dose. We've established the safety of the compound. It's usually greater than 70%. As I mentioned before, the investment at this stage is enormous. That's why it's essentially a very gradual, essentially ascendancy to this level of testing. Again, the cost per patient is very high. Okay. Now I'm going to turn the FDA, how they're involved in a process. I'm not going to spend a lot of time on it, but remember this cliche. They all know the size of your jockey shorts before you're through with this trial. That's very simply how to describe this. They're involved in every step of the way. They know the animal studies. They know the endpoints that you're going to be. They know the statistics. Believe me, they've been there. These are excellent professionals, but it's very much essentially a hands-on type dialogue. This is the way to go. So I'll not go through that. Now, let's turn to something a little bit more interesting. So we're getting out of theoretical to the practical. Where do these drugs come from? And I've isolated this to what people perceive to be discoveries all about, these eureka moments where somebody discovers something and voila, I've cured X, Y, Z. The other is entrepreneurs who have made a successful, principally in licensing, have made a successful go of essentially compounds and what I would call repurposed compounds. These are drugs that are used for hit one receptor, have activity in another receptor if you kind of connect that original schemata I put up before. Now, entrepreneurs. We all know Alexander Fleming essentially discovered penicillin. Alex, in 1928, was the first antibiotic that was essentially on the market. Prior to this, there were chemicals on the market that were essentially anti-infectives, but not antibiotics. There's a difference. They had very deleterious side effects, but essentially they came out of the German chemical industry. They were largely synthetic compounds. Antibiotic, by its very nature, is essentially a natural compound. So penicillin is a fluid that came from a mold that landed on Fleming's petri dish, and it's important to understand this, and inhibited bacterial growth. That's what he observed in his lab. It was ignored for a decade until World War II started coming about, and that's when everybody took it serious after the calamity of loss of life in World War I from infection. So what happened? We know from Fleming's notes, he was not looking at mold. not looking at mold. He was looking at nasal mucus as an anti-infective. If you think about it, it makes sense. You know, you're inhaling bacteria in your nose. Would that essentially inhibit bacterial growth? He was looking at saliva, and he was looking at tear fluid, natural substances that he thought would inhibit bacterial growth. Lo and behold, this is the theory. If you read the discovery of penicillin, I'll talk about this. But it's believed that the mold actually flew in from a floor above his lab and landed on his petri dish. He observed that essentially that particular substance inhibit bacterial growth. That is the theory. I have no idea whether that's true, but that will give you the idea of this kind of serendipitous element that's involved in drug discovery. The old cliche is luck is the byproduct of a prepared mind. He obviously had a prepared mind. Warfarin. I know a lot of people in this room are on warfarin, or were on warfarin, which is used for stroke. How was this discovered? We owe this to a veterinarian by the name of Frank Schofield. What he observed was essentially that certain cattle, and we all know the story, it's rat poison, so I won't bother you with that cliche, but it's still used. But what he observed was essentially cattle were hemorrhaging in fields after eating certain hay. That's essentially what he observed. It took about 25 years later where they actually synthesized and itemized, or essentially identified the compound, which we know today is Coumadin, which is still used for stroke prevention. It's named after the Wisconsin Alumni Research Organization, WAR, which is where Schofield was essentially a member of the University of Wisconsin. Okay. Steroids, something we commonly take for example. Here's a quick example. Edward Kendall deserves the Nobel Prize for this. Succinctly, what he observed in 1929 was essentially women who were pregnant lost symptoms of rheumatoid arthritis. So he observed that and he said, what the heck is going on here? What he did was essentially search or examined the urine from these people, identified steroids. It took several years later to essentially synthesize that particular compound for its use today as we currently have. So again, another eureka event. So entrepreneurs. Here's an interesting situation for someone who's a baseball fan. Who's a baseball fan? Anybody? Who you know, do you know the name of the Kansas City Royals baseball field? It's called, this is the guy that founded that team. Now, what's interesting about this particular guy, he had no scientific background, none. He was educated in a community college and essentially went on to, I'm particularly familiar with this compound called diltiazem. I am on it right now. Many of you may be on it, or sister compounds that emanate from this. But what happened in this situation, he took the risk, which I might say a Japanese company was developing this and essentially said, well, I'm going to buy this compound and rights. Nobody else in the United States wanted it, including my company. Our licensing department didn't want it. And the reason for that was the mentality, getting back to essentially, Tanabe's a Japanese company. The mechanism of action of this particular compound was not familiar to cardiologists or people involved in cardiology research. So it was rejected out of hand. They didn't know what this receptor was. The prevailing view were beta blockers, which I cited before, which predated diltiazem's development, were the accepted theory of how to control high blood pressure. What ended up essentially that Japanese clinical data. He eventually developed the drug, and essentially amlodipine, if anybody's on that particular product, whoever, this is related to Kaufman's discovery. Kaufman essentially made millions of dollars based on that particular drug, and essentially sold the company for in billions of dollars to essentially another pharmaceutical company. Another interesting story is Sam Waxel. This is perhaps somebody you might know. He founded Imclone. The interesting twist to this story is he licensed a compound from Eli Lilly to treat colorectal cancer. He had an insight that essentially, so he didn't discover anything. And I think if you look at the history of Bill Gates, you'd find out that he actually licensed in the iOS system, but that's another story. But essentially what this guy did, he says he recognized essentially that this compound could have utility. So he bought it from Lilly for about $50,000 to $100,000. Essentially what happened, he essentially developed this, and essentially Imclone sold back to Lilly for $6.5 billion. So, of course, Waxel, along with Martha Stewart, were guilty of essentially insider trading. Now, repurposed drugs, thalidomide, just as I mentioned in the introduction, this is a compound I'm certainly our generation is quite familiar with, but essentially was never approved in the United States, caused a number of birth defects. It was developed by a German company, and very simply was not approved for marketing in the United States based on a heroic stand by the FDA. Now, how was this drug was repurposed? In other words, it was used for a different indication. It's now on the market, and the drug of choice for multiple myeloma. Now, let's just think of it. This is a licensing idea that was brought into me personally. And I just said to the fellow who presented it to me, I'll need title insurance if I license this insurance for my own title, because I was going to get flushed out based upon the history of this compound. This compound had a series of issues with birth defects. Nobody was going to go near it in terms of its predominant side effects. And if you think about the clinical trial management here, this disease is fairly prevalent among women. So how do you control for essentially, how do you administer birth control to the women who are premenopausal to avoid essentially unforeseen side effects with thalidomide given to them? So the complexity of this in terms of its clinical development was pretty austere. Essentially, you people in Summit have benefited from this, whether you know it or not. The company was headquartered up to recently here in Summit. Celgene, acquired by Bristol Myers. Okay. Finally, cyclosporin. This was discovered in basically, it was a fungus discovered in Norway by this scientist. He had no idea what to do with this compound. It essentially is immune suppressant. Long story of this is that he found a use for the compound in reducing the probability of organ rejection. This compound made modern organ transplant possible. By suppressing the immune system, your own immune system is not recognizing an organ that is transplanted into your body for essentially, that's a foreign item. Okay. Cyclosporin is now used for dry eye, so it had different uses. Organ transplant is also used for, but it's marketed as Restasis. It's used for dry eye because that's an autoimmune disorder. Now, getting back to what Mark said, I also have a repurposed drug, oxybutynin, which I essentially recognized and patented. We had discussions with Merck and Novartis on this compound, So instead of my residence in Monaco, I'm looking for a trailer park in New Jersey. So anyway, the long and short of it is that these hit two similar receptors, and basically Botox, we might realize was used for wrinkles and now has migrated into migraine pain. Viagra for basically was originally thought to be a cardiovascular medicine, now an ED. Aspartame, a common sweetener you see at tabletops, was originally developed as essentially a cardiovascular agent. And the scientist tasted it with his finger and said, voila, it's sweetener. That's actually what happened. Benadryl, you may know, essentially it's used for allergies. It's also the prime ingredient in certain essentially sleep aids like Somonax. So essentially you're marketing the product for its side effects, not its efficacy. So that's what I would characterize as repurposed drugs. And of course, mine is yet to see the light of day. So one last question, and I'll end this quickly. Medicare, the Inflation Reduction Act, how is it going to affect essentially drug discovery? There's essentially the provisions in it. I'm not going into a lot of detail here, but there's direct negotiations between RX companies and Medicare on specific products. There's 10. It goes into effect in 2026. Selected drugs are indicated for price reductions. New drugs allowed 7 years of exclusivity if you're oral, 13 years if they're proteins. Again, this will essentially shift pharmaceutical resources into developing proteins, which was my earlier point. Now, what's the problem with this? Sure, everybody gets the free lunch, drug prices are going to be reduced. But the issue that involved here is that the companies are operated not on a product by product basis, but essentially a portfolio of research. So what's going to happen is that new drugs that come out are launched, price increases are going to be more than expected. And why is that the case? Well, the new drug will have 9 years or 13 years of exclusivity before negotiations with Medicare, and essentially will make up for that loss of revenue in the out years by basically raising prices now. In addition, the prices will go up for existing compounds outside the purview of Medicare, offsetting some of the price decreases. So that's kind of the way the system works. Later stage trials in this area are going to be terminated. The way oncology drugs are basically developed is you have an initial indication, let's say it's bladder cancer, and then you go on and pancreatic and this, that, and the other thing. What's going to happen as you approach this 9 or 13-year time horizon, it'll take two years to do a study, the studies are going to be essentially dropped. So in the outer years, you're going to have less interest in developing additional indications for compounds. And the best illustration I could make is in oncology, which I think we all have a stake in. Certain drugs will basically be marketed to the non-Medicare class. The most fitting example of this is, for example, we're all familiar with diabetes drugs, which have this dual indication of essentially treating diabetes, kidney disease, and essentially weight loss. Well, weight loss is a problem for everybody, but it's probably more of a problem for younger people outside the Medicare system. So that's essentially the way that's going to work. It's going to be shifting priorities outside of that Medicare umbrella where possible. It's not always possible, but where possible. Finally, new drugs will basically take on new delivery systems. The drugs that come under the Medicare price negotiations will be in one form. In the case I've cited here is Keytruda, which is essentially about 50% of Merck's sales now, and essentially is marketed as an infusion. They are working on essentially a subcutaneous injection. So you're going from essentially the administration of a drug in a back office to essentially probably or essentially a nurse doing an injection in your arm in outpatient. So they changed the composition of the compound. They're out from under essentially Medicare price reductions. You sure will reduce the price on the old medicine, but it's not being used terribly well. So you can have that, but we're going to go ahead with this. So that's kind of the strategy. May not work with all drugs. This is not a simple exercise to do. It's not like putting shaving cream that was originally an 11-ounce can in a 10-ounce can. It requires basically bridging studies, which basically say this compound in this new delivery form is equivalent to the old delivery form. So generally speaking, blood levels are the criteria for those approval, but it could get dicey in the area of cancer. So I'm going to kind of just summarize this with, we've learned that licensing or discovery, the sources of this, the lock and key concept, which I hope you take home and understand, the development process, which we talked about, the objective of research to hit primary and secondary indications, the types of drug discovery that's occurred, which I've classified in these broad categories, and essentially important advances in therapy are made through a combination of skills, some of which is luck, some of which is well thought out science, but essentially it kind of works. And you have to give scientists and companies a free hand in developing these things. So that's essentially it. I hope you enjoyed it. So that's it.
Stay up here. Thank you so much, Mike, for that. That was a wonderful presentation. I learned so much. Didn't you all? You did. I know you did. Questions, you know the routine. If you have a question, come up here. Larry, come on right up. We have limited time.
In 2000, my brother was diagnosed with a pretty nasty form of leukemia, and he had a 80% chance of not surviving 5 years. It's 2023, and he's been free of the illness since 2001. That's when he went from the clinical trial back to actually taking the drug. That drug is something called Gleevec.
Gleevec, yeah.
Can you talk a little bit about it, and then talk about what about a generic form?
Well, Gleevec, I think Gleevec is oral. I think it's marketed. So your benchmark for essentially, and every year wants to pay less for products. Your benchmark basically is, if you're on an oral compound, chances at some point will be generic. So that will basically, I'm not familiar with exactly when the patent expires on that, but there's other strategies the pharmaceutical company can impose to keep the pricing level higher. And that is to change the dosing of the compound. So essentially they obsolete essentially the original dose and essentially move on to something else. So the generic company that's trying to knock off, say, Gleevec, which was originally approved in 20 milligram dose, if the company says, well, 30 milligrams are basically going to be the standard of care now, there's essentially a barrier, if you will. It's not insurmountable, but some institutions, Sloan Kettering in this case, would recognize the efficacy of the 20 milligram, go for the generic, and prescribe it. Moreover, in New Jersey and I think elsewhere, the pharmacist has the ability to substitute a generic, whether you like it or not. So in the case of Gleevec, that will be automatically done at the pharmacy. Okay.
Thank you, Mike.
Yeah.
Mono?
Yeah, it's obvious that getting drugs on the market and so on is a hard job, an expensive job. And so the drug companies want to develop drugs that will give them good money back. But what about the simple things that are not being developed so on right now? We're getting into a period, and probably are already, where antibiotics are no longer available for the everyday things. And, you know, all of us get bumps and lumps and infections, 7 days, 10 days maybe, and that's it. So how are we going to solve the problem that no more new antibiotics aren't being developed because it doesn't pay.
Okay, simply speaking, I think here's where the government can play an active role. That if they extend the patent life of these specific substances, essentially that will do the trick. Now, what is the situation with antibiotics today? I'll tell you about a personal story and then essentially the general picture. Well, maybe I'll start with the general picture. Essentially, what happens in a hospital environment, and I get this, hospitals want to essentially control cost. So they'll take a urine specimen or a blood specimen and determine what particular bacteria or virus is affecting you. They will intend say, well, what antibiotic is going to work in this situation? In most cases, it's going to be a generic antibiotic. So the dilemma that the companies face is that if you're talking about competing within a generic, essentially marketplace, where the hospital is going to say, well, I'm going to try drug number 1, that's generic, drug number 2, drug number 3, and then essentially I'll eventually get to you. That's not a particularly appetizing pricing environment to recover the research dollars that's spent. That's simply the fact of the matter. Further, we talked about the size of these plants, the investments required. Antibiotics fall into that routine. Essentially, you're talking about enormous expenses to establish fermentation facilities to produce antibiotics. And that is another barrier as to why indeed most of the products, say 90% are sourced from India and China. So I see essentially the only way it's basically going to be resolved if somehow the government steps in and say, for these particular indications, the patent life on a product will be extended, perhaps. But to be honest about it, I was told at a certain company, don't bring in an antibiotic. We're not going to consider it. Forget it. Just look at something else. So that was essentially it. Finally, the story. We had an antibiotic that was going to be used for upper respiratory tract infections. Essentially, the drug worked, but what the FDA indicated that it wanted a 2-year toxicology study to see how the drug is metabolized. So you're sitting there and say, okay, the drug is going to be used for perhaps after this queue, essentially your 4th or 5th in line, perhaps for 5 days. And I've got to do a 2 years toxicology study with an uncertain outcome. So I think any reasonable person would come to the conclusion, we dropped it. And that's essentially what happened in real time. Okay.
Next is Al Aho online.
I agree.
Hi, thank you very much, Mike, for a fascinating and interesting talk. For a number of years, I've been curious about how drugs are named. How is it done? And can the name of a drug affect its commercial success? I'm thinking of Ozempic as an example.
I never really got involved in trademarks or, the only one I can think of is, for example, Lipitor, lower lipids, which is essentially a carrier for cholesterol. Other drugs, it escapes me why they're named, whatever they're named. And there's some scientist back there that knows the mechanism of action. So I'm not really sure that essentially I understand how these things are named. So I don't know.
Ron Weinger.
Yeah, gotta do that. Drug ads on TV are probably more dominant now than cigarette and beer ads were in the 50s. Do drug companies actually expect the viewers to go to their doctor and say, give me that drug?
Well, the old saying about advertising, I know 50% is wasted, but I don't know which 50%. So getting to the point here, there may be a naive belief that among drug companies that an active campaign to influence consumer behavior they're going to rely. But the problem the drug companies face, when you go to your doctor and you say, geez, I have high blood pressure, the pharmacy benefit manager says, well, I've got one from column A, I got one from column B. Well, that product kind of is the same as the one I have here, and those are generics. So what the pharmaceutical companies are trying to influence is that dialogue. So chances are, if you have high blood pressure, you get metropropyl as your key drug. If you're still on that, you should talk to your physician, because it's probably about 80 years old in terms of, but it suffices. It's good compound, doesn't necessarily mean it's wrong. So the drug companies are trying to influence payers and essentially the consumers.
Bill Tittle online.
Yeah, great talk, Mike. Thank you. The pharmaceutical industry to me seems to be very unstable financially compared to the chemical industry, which is, but it isn't it also, it's not mature at all, is it? I mean, we're learning new stuff all the time about the complexity of the body and all the rest of it. Do you feel that it's a mature industry?
I think it is a mature industry from a standpoint of there's only a certain amount of companies that can afford to address these large markets financially. I cited this cost for developing a drug. Those are real-time costs, $20,000 per patient, because he's to manage those trials. Okay, this is not Pharmaceutical Manufacturer Association press releases. This actually happens. So there are a lot of barriers to entry, but where essentially the innovation is coming from is venture capitalists now actually fund a lot of these university technologies, put a management team together, and essentially start a company and hope they will partner at some point with a pharmaceutical company. So that's the engine of growth. So I characterize it as almost a dichotomy. Large companies are in a difficult situation because of P&L pressures and growth pressures, whereas you have this funding mechanism of new products coming from the venture capital industry, funding university technology, and essentially finding companies and hopefully partnering with large pharma.
DJ Perone.
Thank you, Mike. No, you don't have to. No, you're the expert. This was brilliant. Thank you very much. And I wish I would have heard your lecture 40 years ago when I was in medical school. One question that you touched on is the international collaboration of the pharmaceutical companies. How does the United States stand in the world of new drugs? If I may, also I have a quick second question. It occurs quite frequently. You're at a cocktail party, you have a glass of Chardonnay in your hand, and you have this person who comes to you and said, "Oh, pharmaceutical companies, they are just robbing people." So I know you already answered that, and I think I could answer to that person, counteract their comments. But I'd like to know, on a quick answer, what do you do to that person? So the international situation and the cocktail party situation. Thank you.
Yeah, I think in the international situation, because of the flow of technology, companies are very attuned to the information emanating from different areas of the world. So it's been internationalized from a standpoint of communication on essentially drugs, approaches, what approaches have been working, what haven't been working. So the information flow is there. In terms of clinical trials, however, in the 1960s, essentially, in the case of daltiazem, the data that we had from Tanabe, the Japanese company that owned it, was Japanese clinical data that was not accepted in the United States because of the genetic differences between Japanese and Americans. That has changed. Now you can do a clinical trial. The last trial I had was in 10 countries throughout. So it's being internationalized. The results are being published in local scientific publications. So the flow of information is there. The United States still remains the dominant entity in discovery of new approaches to disease. Not in all cases, but in a lot of cases. Principally the larger companies and the biotechnology companies that I referred to. Now, as far as pricing is concerned, this essentially is a difficult issue. First of all, I talked about the enormous costs that are involved in these studies. And if you handicap the risk of whether you're going to be successful, not every drug, as I indicated, is going to work. So how do you price a drug accordingly? Generally speaking, you will price the drug according to what the competition is out there. So in other words, you'll look at the competition, you'll know that that particular drug that's out there is being used in this setting, diabetes or whatever the case is. And you'll essentially know that that particular drug, which is critical, is being reimbursed by Horizon or CVS, Blue Cross or United Healthcare at those prices. So that's the way it's right. Now, if you have a new indication, it's pretty much at the discretion of the company. I think there is a compassionate element among these companies not to railroad essentially certain patients regarding essentially pricing. But I think that varies from company to company. I know, pharmaceutical companies are viewed as one step above welfare mothers and one step below the oil industry, something like that, depending upon the political atmosphere. But that's essentially what the dialogue is. And pricing is basically, it's not scientific, it's just an art in terms of essentially and a competitive element. Okay, right here.
Okay, we're really running out of time, so we're going to have the last question now from Walt Meissner online.
Yeah, thanks for a great talk. The question I have is, most of these studies last from 1 to 3 years, and they note the short-term side effects, but people end up taking these drugs for 10, 20, 30 years. Is anybody looking at the long-term side effects and possibly wrapping that into modifications of what they have?
Okay, I think there's an excellent question. I think in certain cases where the question, let me just reiterate it because we're a little garbled. A drug is approved, you base it on the clinical information you have at that time. The FDA will dictate whether indeed you should look at this particular class of drugs, whether it interacts with them, or basically what the effects are on a certain cohort of patients. It really goes on and on. But the question he's asking is, what happens after the drug is approved? Are there requirements to basically look at the drug to avoid unforeseen side effects? Okay. What happens is that in the pharmaceutical business, they're all essentially armed with what they call a medical affairs office. So any side effects that are coming in from the field, from physicians, they know where to contact the company. And essentially these things come in, some action is taken, some is not. If there's a proliferation of side effects associated with the drug, they'll take action. They're required to notify the FDA about any safety concerns. So that is addressed through a large organization that has these capabilities. In certain cases where there's ambiguity about the long-term side effects, the FDA will require essentially to say, well, there's an urgent need for this colorectal cancer drug, but we recognize the side effects could be X, Y, Z. We're going to let you have an approval here, but you're going to have to do a study to essentially address these issues out after marketing. So that's an ongoing process, both orchestrated by the company and dictated by the FDA. What is more problematic, which gets into an area where there must be, I would say, 50, maybe, I don't know, 30 hypertension or high blood pressure drugs out there. What degree are there interactions between these drugs, between a generic drug and essentially something that's been approved? The generic drugs are not receiving any support for essentially their research. They're out there, they're a commodity, it's sold on price. Whereas the company comes out with a new hypertension, it's not clear to me how indeed the companies basically monitor those interactions other than through the medical affairs unit within the company. Okay.
Thank you. Thank you again, Mike. I want to present you this certificate, and you know, it has this probably an herbal remedy here on it. We're not going to take a lot of time. We're way out of time now.
Okay, thank you.
I call for the old guard salute